Welcome to the Deep Dive Ortho, the show dedicated to giving you the essential insights you need, fast. As medical professionals, from orthopedic surgeons to residents, you're constantly navigating a sea of information, aiming to make the most evidence-based decisions for your patients. It's a relentless pursuit, isn't it? But what if I told you there's a gaping hole in that evidence base? A vast amount of medical research that remains hidden from public view, directly impacting your daily practice. Today, we're embarking on a crucial deep dive into this unseen evidence. We're talking about clinical trial results and other vital research that never sees the light of day. This isn't just an academic problem. It can distort our understanding of treatments, influence patient safety, and waste precious resources. To help us navigate this intricate landscape, I'm delighted to welcome Professor Mo Imam, an esteemed expert in this critical field.
Thank you for having me. It's a vital topic.
Our mission in this deep dive is clear. We're going to unpack the full scope and profound impact of research waste, focusing particularly on unpublished results. We'll examine how this directly affects not just the orthopedic community, but the wider medical world, and crucially, how greater transparency can revolutionize the integrity of the evidence you rely on every single day. So, let's jump straight in. Professor, let's start with a statistic that I find absolutely staggering. Imagine that roughly half of all clinical trials ever conducted have simply never been reported. For our listeners, busy medical professionals making critical decisions daily, what are the immediate practical implications of a figure like that? It's almost like operating with half the instructions.
It's a truly striking figure, yes, and one that campaigns like all trials have been working hard to bring to light. Based on extensive analysis, it's estimated that, around 50% of clinical trials have indeed never made their full methods and summary results public. Yes. And for you, the clinician, the implications are profound and immediate. Firstly, this creates a really distorted evidence base. When a significant portion of trial results, especially those that might not show a positive outcome, are missing, you just don't have a complete or unbiased picture of how interventions truly work.
Right, you're only seeing part of the picture.
Exactly. You might be seeing only the successes or, a skewed version of reality. For an orthopedic surgeon, imagine evaluating a new implant or perhaps a surgical technique. If trials showing no benefit or maybe unexpected complications are buried, your understanding of its true efficacy and safety is fundamentally incomplete.
Which must make decision-making incredibly difficult.
It directly leads to poorly informed treatment decisions. You are then forced, essentially, to make choices based on partial or even skewed information, which can, as you can imagine, pose unacceptable risk to your patients. You might be opting for a treatment that is less effective than you believe it is, or unknowingly exposing patients to risks that were actually identified but never reported. This isn't just about efficacy. It's fundamentally about patient safety.
And what about the impact beyond the clinic?
Well, beyond that, there's a significant loss of public trust. Patients volunteer for trials, often taking risks, believing their contribution will advance medical knowledge.
Of course.
When results are withheld, it's a betrayal of that trust. And it can understandably discourage future participation, which harms the entire research ecosystem.
And the resources involved.
Absolutely. Let's not forget the tremendous waste of resources. Think of the economic cost, the human cost, of designing, recruiting for, and conducting these trials. Time, effort, often public funds only for the findings to be locked away. It's an egregious waste of investment.
It feels unethical.
It is. Ultimately, there's a fundamental ethical obligation to report results, given the risks taken by trial participants. It's non-negotiable, really.
The ethical weight of this is immense. It truly puts the gravity of this issue into perspective. But when we hear a figure like around half, it does raise an immediate question.
Yeah.
How robust is that statistic? How do researchers even begin to quantify something that's, hidden?
That's a crucial question, yes. And it's important to understand how we arrive at this estimate. The around half figure isn't just plucked from thin air. It's consistently highlighted by major advocacy groups. And, more importantly, it's derived from a series of robust, systematic reviews.
Okay, so multiple studies looking at this.
Exactly. For example, a major analysis by Song and colleagues back in 2010, which was actually funded by the NHS, pooled data from hundreds of individual studies. These covered tens of thousands of trials.
Right.
And this comprehensive review consistently found that, yes, approximately 50% of clinical trials had simply never reported their results.
And is there a pattern to which results get reported?
Yes, and that's particularly concerning. What those findings showed, and what's been reinforced by other research since, is a significant bias towards positive results. Trials showing a statistically significant or, positive outcome were found to be 2 to 3 times more likely to be reported than those with negative or null findings.
So the published literature is inherently skewed towards success.
Precisely. It creates an artificially optimistic view of interventions because the failures or the no effects are disproportionately missing.
Now, have things improved recently? We hear a lot about pushes for transparency.
It's vital to recognize that while we have seen some improvements in publication rates for new trials recently, which is good news, this doesn't erase the massive, persistent backlog of unpublished trials stretching back decades. Many of these older trials concern treatments that are still in widespread use today.
So the drugs or procedures we use now might be based on evidence that's incomplete because of this historical issue.
That's exactly the concern. Even if future trials are better reported, we still have this huge historical deficit affecting the very treatments you might prescribe or procedures you might perform today. Quantifying this perfectly is challenging, of course. There isn't one single master list of all trials ever conducted, and definitions of reported can vary slightly.
Right.
But these large systematic reviews provide our best and, frankly, quite sobering estimate of the scale of the problem.
That context about the difficulty of measurement and the historical backlog is really insightful. It's clearly not just about current practices, but a deep-seated problem with historical roots. Could you give us a sense of the specific evidence that underpins this understanding? What have researchers actually uncovered about the prevalence of unreported trials over the years?
Certainly. There have been multiple studies over time that have consistently painted a clear picture of this pervasive problem. It's not just a theoretical concern. For example, some early investigations looked specifically at clinicaltrials.gov, which is the world's largest clinical trials register. Ross and colleagues, for instance, published a study showing that even among trials registered there, less than half had results reported on the platform at that time.
So even registration didn't guarantee reporting?
Not necessarily, no.
No.
And later studies highlighted ongoing issues with compliance, even when legal mandates were in place. Prail and his team, again looking at clinicaltrials.gov, discovered that only about a fifth, just 22% of trials, actually met the legal requirement in the U.S. to report summary results within 1 year of completion.
Despite it being law?
Despite it being law. It was a stark demonstration of the gap between regulation and actual practice on the ground.
And what about larger trials? Does size make a difference?
You might think so, but Jones and colleagues focused on larger, arguably more impactful studies, substantial randomized clinical trials involving over 250,000 participants collectively. They found that even among these, a considerable percentage, nearly 30%, remained unpublished. That's a huge amount of participant data, essentially hidden from view.
That's astonishing.
And then a very comprehensive systematic review by Schmucker and others pooled data from dozens of studies, over 20,000 trials in total. They reiterated that figure. Only about half of trials were published in journals. And critically, they confirmed that positive findings were about 3 times more likely to appear in print.
The positive bias again.
Exactly. And another critical finding came from Anderson and colleagues, who looked at reporting rates over longer periods, up to 5 years. They found an interesting difference based on funding. While industry-funded trials eventually reached a higher reporting rate, around 80% by 5 years, government or academic-funded trials lagged significantly behind, with rates closer to 40–45%.
Why would that be, do you think? More regulatory pressure on industry?
That's likely part of it, yes. But these various studies taken together provide a consistent and frankly compelling body of evidence for widespread non-publication and persistent reporting biases across the entire medical research landscape. It's not just one study saying this, it's a pattern.
Those figures really do put the scale of the challenge into perspective. It's clearly not a small, isolated issue. Professor, you've alluded to the broader concept of research waste beyond just unpublished trials. Could you help us understand what this wider concept truly encompasses? It sounds like it goes far beyond simply not reporting results.
Absolutely. While unpublished trials are a critical piece of the puzzle, research waste is a much broader and more systemic problem. It affects the entire life cycle of research right from the very beginning. We generally define it as our poor quality research output that is often perceived as of minimal use to health policymakers and clinicians.
Minimal use, so research that doesn't actually help.
Essentially, yes. It's about where value is lost along the way, from the initial research question right through to the final dissemination or lack thereof.
And what's the scale of this broader problem?
The scale is truly stark. Beyond the, over 50% of research that is simply not published, another significant portion often suffers from avoidable design flaws. Poor methodology, basically.
Right.
And even among what is reported, a substantial percentage is either unusable, perhaps the reporting is so poor you can't figure out what they actually did, or it's incompletely reported.
And the financial implications.
If we were to try and quantify this financially, estimates suggest we're talking about approximately 85% of global research investment being lost due to these kinds of correctable flaws.
85%.
Yes, it translates to billions of pounds globally that are essentially wasted. Funds that could have been used to genuinely advance patient care, develop new treatments, improve orthopedic outcomes, but instead are lost in this system.
Billions lost due to correctable flaws. That's a staggering figure. What are the root causes then? If it's 85%, it must be woven into the very fabric of how research is conducted rather than just one single problem point.
Indeed, it's a multifaceted problem. Waste can occur at nearly every single stage of the research process. It's not a single bottleneck, but more like a leaky pipe with holes all along it.
Okay, so where do the leaks start?
Well, one primary cause is the lack of prioritization. Often, research is undertaken without a thorough assessment of genuine evidence gaps, or without really addressing the most pressing clinical needs.
Like doing another minor variation study when a big question remains unanswered?
Precisely. Conducting another trial on a slightly varied orthopedic technique when there are already dozens of similar studies, while perhaps a critical question about long-term implant failure remains unanswered, that's a form of waste. Closely related are imbalanced research questions that are poorly framed, or don't adequately consider the existing evidence, potentially leading to redundant work from the start.
And then the research itself.
Yes, then we have flawed study design and execution. Methodological weaknesses, whether in selecting control groups, randomization, blinding, follow-up. These can compromise the validity and reliability of the findings, rendering the entire effort questionable, no matter the result.
Can you give an orthopedic example?
Sure. Imagine a poorly designed trial comparing two hip replacement approaches where patient selection criteria are far too broad, mixing different patient groups, or where follow-up is inconsistent, losing many patients. The results would be hard to trust. The use of unvalidated outcome measures is another significant contributor.
Meaning we're not measuring the right things.
Exactly. If we're not measuring what truly matters to patients, say, long-term functional recovery or quality of life, rather than just short-term radiographic changes, the results might look good on paper, but have limited clinical utility for you and your patient.
Okay, so the research is done, maybe not. What happens next where waste can occur?
Even when research is well conducted, waste occurs if it becomes inaccessible. This includes non-publication, obviously, but also findings buried in proprietary databases or hidden behind expensive journal paywalls.
Right.
Publication and other reporting biases are rampant, as we've discussed, particularly to focus on positive results or selective reporting of only favorable outcomes, which distorts the evidence base.
Selective reporting. Choosing which bits to publish.
Yes, sometimes even within a single study. We also see significant duplication of studies. Researchers might embark on new trials without thoroughly reviewing previous findings, perhaps because those findings weren't published or easily discoverable, unknowingly replicating work that's already been done or has already shown no effect.
Which is linked to trial registration.
It is. Insufficient registration of clinical trials, or registering them too late, contributes to this duplication by making it difficult to track what research is ongoing or already completed. And finally, there's the subtle but insidious issue of spin in interpretation.
Spin? You mean how results are presented?
Exactly. Where authors or sponsors perhaps distort results, maybe through careful wording or selective emphasis, to present them more favorably than the data truly justifies. This also contributes to misleading the medical community. It's a complex web, and as GM Sogi articulated it quite powerfully, this situation is economically, scientifically, and ethically unjustifiable.
It's clear this isn't just an abstract academic problem. It has real, tangible consequences for patients and the allocation of vital resources. I know there's a growing recognition of this problem, which is encouraging. Could you tell us about some of the concerted efforts being made to tackle this issue systemically?
Absolutely. The growing recognition has indeed led to significant calls for action and some important initiatives. A notable example is the Reduce Research Waste and Reward Diligence Alliance, often shortened to the Reward Alliance.
Reward, okay.
This alliance was actually founded following a conference in Edinburgh back in 2015. What's key is that it brings together a really wide range of stakeholders, researchers, institutions, funding bodies, journals, regulators, even patient groups.
So everyone involved in the process.
Precisely. Their goal is to collectively tackle these systemic issues across the board. It highlights that no single entity can solve research waste alone. It requires a concerted effort from everyone involved in the research ecosystem to implement changes at every stage, from setting priorities and designing studies better, right through to ensuring proper conduct and transparent reporting. It's about making sure that the immense investment in biomedical research actually yields valuable and usable knowledge for you, the clinician, and ultimately for patients.
That's a vital alliance, recognizing the complexity and the need for a multi-pronged approach. Now, let's delve deeper into one of the most persistent biases you mentioned, this preference for positive findings. Why do studies with non-significant or negative results so often remain hidden in that proverbial file drawer? What's driving that?
It's a deeply ingrained issue, unfortunately, rooted in the historical culture of research and publishing. Journal preferences have historically played a significant role. Well, think about printed journals. They had physical space constraints. This naturally led to a preference for positive or novel findings, results that were perceived to tell a more compelling, more publishable story, something new and exciting.
But we have online journals now. Surely space isn't the issue it was.
You're right, physical space is less of a constraint, but that cultural bias has proven remarkably persistent. Even with online formats, editors and reviewers often still look for interesting or groundbreaking results. There's a perception, right or wrong, that positive findings generate more interest, more citations.
And this leads to researchers not even trying to publish negative results.
Exactly. This leads directly to what's famously known as the file drawer problem. Non-significant results are often just, well, filed away sometimes literally, and never submitted for publication. Researchers, quite understandably, need to publish to advance their careers, particularly early career researchers. If they perceive their negative results as less publishable, or perhaps even fear criticism for them, they might simply not attempt to submit them. This creates that skewed perception of intervention effectiveness across the published literature, where only a fraction of the full story is ever revealed.
Is it also just harder to write up negative results?
There's an element of that too, yes. There's often a perceived difficulty in interpretation of negative results. It can be harder to explain a lack of effect convincingly than a clear positive one.
Why is that?
Well, the question always arises, was there truly no effect? Or was the study perhaps flawed in some way? Was it underpowered? Did it use the wrong patient group? Was the intervention not delivered properly? Was it exploring the wrong parameter space? This inherent ambiguity can make these results harder to sell to editors and reviewers, who might perceive them as less impactful or less definitive.
So they're seen as less interesting.
Potentially, yes. As one academic described it, it's a lot harder to show that a negative result is a valid result rather than you screwing up in some fashion. This perceived lack of interest, coupled with the ambiguity, becomes a significant barrier, especially in competitive fields like orthopedics, where novel techniques or devices often grab the headlines.
It really highlights the systemic pressure points within academia and publishing. When results are selectively published like this, it feels like we're only getting half the story, maybe less, and that could have serious consequences for patient care. What are the specific ethical implications of this selective publication?
The ethical implications are profound, particularly for you, the clinician making decisions, and of course for your patients. Hiding negative results means that the medical community, including yourself, receives an incomplete and potentially quite misleading understanding of an intervention's true efficacy and safety profile.
So decisions are based on faulty intelligence, essentially.
In a way, yes. You might be making decisions about a patient's care, say, recommending a specific surgical approach, perhaps a new type of knee implant, or prescribing a medication based on what appears to be a strong positive evidence base in the published literature, when in reality, numerous negative trials showing no benefit, or perhaps even indicating harm, are simply not in the public domain. They're in that file drawer.
Which completely changes the risk-benefit calculation.
It completely distorts the risk-benefit analysis for any given treatment. This isn't just about striving for good science. It's about fundamental patient safety and the integrity of the medical profession.
Are there official stances on this?
Yes, very clearly. The Faculty of Pharmaceutical Medicine, for example, put it powerfully. It is unethical to withhold publication of any results of research on any pharmaceutical product, whether the results are positive, negative, or inconclusive. That really underscores the moral imperative. Transparency isn't optional here.
That statement really drives home the ethical imperative. And it brings up an important point. And you mentioned earlier that it's harder to interpret negative results. Does that mean not all negative results are created equal? Some might truly be uninformative, while others could be incredibly valuable. How do we distinguish between these?
That's a critical distinction, and you're absolutely right. We shouldn't necessarily advocate for publishing every single failed experiment, regardless of quality.
So what's the difference?
Well, on one hand, you have poorly conducted or genuinely underpowered studies. These are trials with significant, unavoidable design flaws, perhaps insufficient statistical power to detect even a meaningful effect, or maybe inadequate analyses. If a study is fundamentally unsound, for instance, an orthopedic trial comparing two techniques with a tiny sample size that inevitably fails to show a statistically significant difference, its non-significant results may indeed be uninterpretable.
So those might not add much value.
They might not merit standalone publication because the negative result is likely just noise or a consequence of poor design. Publishing them without heavy caveats could even be misleading. It's about the quality and validity of the finding.
And the other type?
On the other hand, we have well-conducted, informative null findings. These are studies that set out to thoroughly investigate a clear, relevant hypothesis, are appropriately powered to detect a meaningful difference if one exists, and are meticulously executed. When these studies demonstrate a genuine lack of effect, that's incredibly valuable information.
Especially if it challenges current practice.
Exactly, especially if it contradicts conventional thinking, or perhaps previous smaller, less robust studies, or challenges widely held assumptions about a treatment. For instance, if a large, robust orthopedic trial shows definitively that a new, expensive surgical technique offers no benefit over an existing, less invasive standard of care.
That's crucial information.
It's incredibly important information for surgeons, for patients, for guideline developers, for healthcare funders. It prevents wasted resources on the new technique, avoids potential unnecessary risks from a new procedure, and helps refine clinical practice guidelines based on solid evidence. These types of null findings absolutely merit publication and wide dissemination.
So it's about the quality of the study, not just the direction of the result.
Precisely. And even when null findings aren't perhaps the main focus of a paper, incorporating what you might call negative tidbits or descriptions of blind alleys explored during the research, perhaps in supplementary materials, can be immensely helpful.
How so?
Well, detailing, for example, particular antibodies that didn't work in a lab study, specific drug concentrations that had no effect, or certain surgical approaches that were piloted and abandoned due to unforeseen complications sharing. This prevents other researchers from unknowingly going down the same dead ends. It saves enormous amounts of time and resources. This is particularly valuable in fields like orthopedic device development or the refinement of surgical techniques, where trial and error is very common.
That's a pragmatic and important distinction, ensuring we value truly informative null findings, but don't necessarily clutter the literature with poorly executed studies. Moving on, Professor, the All Trials campaign has clearly been a significant force in advocating for this transparency. Could you elaborate on the progress made through such advocacy and the legislative changes it has helped to bring about globally?
Indeed, the All Trials campaign and similar initiatives have been absolutely instrumental. Formed back in 2013 by key international organizations, it's gathered broad support, over 95,000 individual signatures, and the backing of hundreds of organizations worldwide. It's become a powerful, collective voice.
And has it led to actual changes in law?
Yes, it has significantly contributed to legislative changes and a generally increased expectation of transparency surrounding clinical trial registration and reporting across the globe. It's raised awareness and put pressure on regulators and policymakers.
Let's focus on the situation here in the UK. What are the latest developments in terms of legal mandates for clinical trial reporting here? Have we seen concrete action?
In the UK, yes, we've seen some very significant recent developments, marking what really feels like a genuine turning point. Following an announcement by the MHRA, the Medicines and Healthcare Products Regulatory Agency, in March 2023.
Okay.
The Medicines for Human Use, Clinical Trials Amendment Regulations 2024 were formally signed into law in 2025. This is a major step.
And what does this regulation require?
It makes it a statutory requirement, effective from the 10th of April 2026, for all medicinal clinical trials conducted in the UK, all of them to be registered prospectively in a World Health Organization, WHO, public register. And crucially, it mandates the publication of a summary of results within 12 months of the trial's completion,
Regardless of the outcome.
regardless of the outcome. This creates a much more robust legal framework. Of course, its ultimate effectiveness will depend on consistent monitoring and enforcement by the HRA, the Health Research Authority, and the MHRA. But legally, the requirement will be there. This means for you, the clinician, future drug trials conducted here should have a much clearer and more complete evidence trail available publicly.
That's a clear legal step forward for the UK. How does this compare to efforts in the United States? They've had mandates for a while, haven't they?
They have, yes. The United States introduced requirements much earlier with the FDA Amendment Act of 2007, often called FDAAA. This law mandated sponsors of many clinical trials, applicable trials, as they define them, to register them on clinicaltrials.gov within 21 days of enrolling the first patient, and importantly, to report results, typically within 12 months of completion.
So similar requirements, but earlier?
Similar requirements, yes, but significantly earlier. However, the major issue in the U.S. was that initially this law wasn't very effectively enforced. There were widespread reports of noncompliance for many years.
Ah, so the law existed but wasn't biting.
Exactly. The good news, though, is that things seem to be improving significantly. A 2023 report by the U.S. Government Accountability Office, the GAO, noted significant progress. Agencies like the NIH and FDA have become much more active in enforcing the rules, actively pursuing researchers and sponsors to bring them into compliance. So while the law existed for a long time, effective enforcement seems to be a more recent and welcome development leading to better transparency in recent years.
And what about the European Union? Are they seeing similar standardization efforts across member states?
Yes, the European Union has also made significant strides towards standardization and transparency. From the 31st of January 2023, all initial clinical trial applications across the EU must now be submitted via the Clinical Trials Information System, or CTIS.
A single system for all EU trials.
Exactly. This system has become the single entry point for the submission, assessment, and supervision of clinical trial data. The aim is to streamline the process, enhance transparency, and make information publicly accessible through a single portal. It's a major harmonization effort.
So we're definitely seeing positive legislative and regulatory movement in major regions like the UK, US, and EU. That's encouraging. But despite these advances, it sounds like there are still persistent gaps and challenges that need to be addressed. What more still needs to be done? What are the remaining hurdles for achieving full transparency, particularly for you, the practicing clinician trying to use this evidence?
That's right. Despite this significant progress, we're certainly not yet at the goal of having 100% of trials being registered promptly and 100% of results being reported within that 12-month time frame. Not even in these highly regulated regions, there are still considerable gaps and ongoing challenges.
Such as?
Well, for instance, while enforcement is improving, as we discussed, we still don't have 100% compliance. Studies continue to show that a significant percentage of trials, even those funded by major bodies like the NIH in the U.S., are still late to be registered.
Meaning the information isn't there when it should be.
Precisely, which hinders transparency and the ability to track ongoing research. Furthermore, untimely result submissions remain a persistent problem. Reports, for example, from the HHS Office of Inspector General in the U.S. indicated that only about half of NIH-funded trials actually submitted their results on time in recent years, often citing insufficient monitoring and enforcement by the funding agency itself as a contributing factor.
So enforcement remains key.
Absolutely. But perhaps an even bigger challenge, especially for treatments already in use, is what we might call the dark age of buried trials.
Or dark age.
Yes. While these improved registration and reporting rates are a relatively recent phenomenon, largely spurred by international policies introduced around 2005 by groups like the ICMJE,
Right.
the vast majority of clinical trials conducted on treatments that are prescribed today actually predate these changes. We're talking about a massive historical backlog of older, often unregistered and therefore untraceable, and unreported results from earlier golden ages of drug development or surgical innovation.
So the evidence for things we use routinely might be incomplete.
It's highly likely. Uncovering these older trials, finding their data, is a monumental task, perhaps even impossible in many cases. And their absence profoundly affects our true understanding of the long-term benefits and risks of many commonly used treatments, including many established orthopedic interventions. You simply cannot assume that just because a drug or device is on the market today, all of its supporting trial data is transparently available in the public domain.
That's a deeply concerning thought.
It is. And finally, we must acknowledge the significant global disparities. While the US, UK, and EU are making these efforts, compliance and research practices vary enormously worldwide. Some regions, unfortunately, show very poor adherence to basic registration policies. Research has indicated extremely low registration rates for trials reported in journals originating from certain parts of the world.
So the problem is much worse in some places.
Yes, it highlights that while progress is being made in some key areas, it's far from a universal solution. A truly global, comprehensive, and unbiased picture of medical evidence remains elusive. There's still a very long way to go.
That global disparity is a critical point, reminding us how much work is still needed internationally. And the idea of a dark age of buried trials really resonates, especially for clinicians evaluating long-standing treatments they learned about years ago. This brings us neatly to a less conventional but increasingly important area for finding information: gray literature. Professor, could you introduce us to this concept and explain why it's considered a vital, yet often overlooked, component of the evidence base?
Certainly, when we as clinicians or researchers think of medical evidence, our minds often go straight to peer-reviewed journal articles published in major medical journals. That's the traditional gold standard, isn't it?
Yes, absolutely.
However, to truly build a comprehensive, unbiased evidence base, we really must look beyond these traditional publications to what we collectively term gray literature. The term refers to material that is either unpublished or has been published in non-commercial form.
Non-commercial form.
Yes, essentially meaning it falls outside the conventional commercial publishing channels like journals and books. It encompasses a vast array of documents, data, reports, and other materials that can hold critically important insights but aren't found in the usual places.
So it's material that might not go through that traditional academic publishing route. Who typically produces this kind of material, and what unique value does it offer to medical professionals like our listeners who are trying to piece together the full picture?
Precisely. Gray literature is produced by a really diverse range of entities. You'll find it generated by individual researchers and practitioners, certainly, but also very significantly by charities, nonprofit organizations, professional associations like surgical societies, for instance.
Okay.
Also, universities producing theses and reports. Government bodies, think of reports from the Department of Health or guidelines from nice international bodies like the WHO or the World Bank. And crucially, patient groups often produce reports or surveys.
A very wide range of sources then, and its unique value.
Its unique value is multifold. Firstly, as we've hinted, some material is only available as gray literature. Think about preliminary research results shared confidentially with funding bodies. They might never be published formally, but contain early signals.
Right.
Secondly, it's often rapidly produced and disseminated. Conference proceedings, abstracts, or posters, for example, might contain key results from an orthopedic trial presented perhaps a year or more before they finally appear, if ever, in a peer-reviewed journal. This offers much earlier access to emerging findings.
Faster access, that's key.
Definitely. Thirdly, gray literature can often provide fuller detail than journal articles. Because it's not constrained by strict journal word counts or page limits, reports or theses can offer much more comprehensive descriptions of methods, more detailed results, or more nuanced discussion.
More depth.
Yes. It also enables diverse viewpoints to be heard. It provides platforms for voices that might be less likely to publish in traditional academic journals, such as patients reporting on their lived experiences with a new knee replacement or the side effects of medication. It also offers valuable industry or sector-focused insights, perhaps from a practitioner's perspective, on implementing a new guideline, which academic journals might not always capture. And importantly, linking back to our earlier discussion. Yes, perhaps most crucially in the context of our discussion today, gray literature plays a vital role in counteracting publication bias. Because it's less subject to the positive results filter of many journals, it is significantly more likely to include reports of negative results or inconclusive findings.
Ah, so it helps balance the picture.
Exactly. By actively searching this realm, looking at trial registries, conference abstracts, theses, government reports, you can potentially gain a much more balanced and complete understanding of an intervention's effects, helping to mitigate that distorted evidence base created by selective publication in journals.
That truly broadens our understanding of what counts as evidence. So, Professor, for our orthopedic surgeons and residents who are already swamped with information, what's one practical way they could start leveraging gray literature, perhaps a specific type, to get ahead or make a more informed decision this week? Something actionable.
That's an excellent question, and a practical approach is absolutely key here. If I had to pick one starting point that offers high value, I'd strongly recommend getting familiar with and actively searching trials registers.
Like clinicaltrials.gov.
Exactly. Clinicaltrials.gov, the WHO International Clinical Trials Registry Platform, ICTRP, search portal, the UK's Clinical Study Portfolio. Search. These are all technically forms of grey literature. And they're increasingly where trial registration and, importantly, summary result reporting are mandated.
So searching these directly.
Yes. By searching these directly for a specific intervention, maybe a new bone graft substitute you're considering, or a novel surgical tool, you can identify ongoing or recently completed trials whose full results may not yet be published in journals or may never be. Crucially, you're much more likely to find listings for trials with both positive and negative or inconclusive findings here, giving you a much more balanced perspective long before it might filter into conventional wisdom or even journal publications.
And they might link to more detail.
Often, yes. Many register entries provide direct links to study protocols, which give methodological details, and some are now required to post summary results directly onto the register within 12 months. So it can be a rich source of relatively unbiased, up-to-date information. It's a powerful first step into gray literature.
That's a fantastic, actionable tip, but you mentioned the lack of peer review. That obviously raises questions about quality. How do clinicians navigate the potential pitfalls of variable quality and authenticity when using gray literature? How can they evaluate its reliability effectively?
That's a very important point, and it absolutely requires a degree of caution and critical appraisal. You can't just take gray literature at face value in the same way you might approach a peer-reviewed article from a top journal. The main challenge is indeed its variable quality.
Because there's no formal review process.
Precisely. Without that formal, independent peer review process, the scientific rigor, the methodological soundness, and the overall reliability of the material can differ enormously. You might also encounter authenticity concerns more frequently. Basic information like the author's credentials, the date of publication, or even the specific publishing body might be missing or unclear, making it harder to verify the source or assess potential conflicts of interest.
So how do you approach it then?
You need a structured approach to evaluation. While there's no single perfect method, I often recommend using the ACITES checklist as a helpful framework or memory aid. It was developed by librarians at Flinders University in Australia.
ACTS. What does that stand for?
Stand for authority. Who actually produced this document? What are their credentials or affiliations? Is the organization reputable in this field? Accuracy. Does the information seem factual? Does it align broadly with other known evidence? Is the methodology transparent and clearly explained, even if not peer-reviewed? Are there obvious errors? Coverage. What is the scope of the information? Is it comprehensive enough for your needs, or is it very selective? Does it acknowledge limitations? Objectivity. Is there any apparent bias? What is the clear purpose of the document? Is it informational, or perhaps promotional, or persuasive? Are conflicts of interest declared? Date. How current is the information? Is it still relevant to contemporary practice, or is it outdated? Significance. Ultimately, how important and relevant is this piece of information to your specific clinical question or decision?
Authority, accuracy, coverage, objectivity, date, significance.
Exactly. By mentally running through these criteria, you, the medical professional, can critically assess the credibility and potential utility of a piece of gray literature. It allows you to extract potentially valuable insights while maintaining a necessary and healthy level of skepticism about its provenance and rigor.
That ACODS checklist sounds like a vital tool for navigating this complex, often murky landscape. Now, let's broaden our perspective again to the wider movement of open science, which seems to have emerged as a direct response to some very fundamental issues in the way research is traditionally conducted and shared. Professor, what exactly is open science, and what core problem is it trying to solve?
Open science is a really broad and important movement that has gained significant momentum over the last decade or so. It emerged fundamentally as a direct response to what's often been termed the replication crisis or reproducibility crisis.
Replication crisis.
Yes. This refers to the widespread and frankly pervasive failures to reproduce the published findings of scientific studies when other independent researchers try to repeat them. This has been particularly evident in fields like psychology and preclinical biomedical research, but the concerns are relevant across the life and social sciences, including medicine.
And why is that failure to replicate such a crisis?
Because if published results cannot be reliably replicated, it severely undermines the credibility and trustworthiness of scientific knowledge itself. It makes it incredibly difficult for clinicians like yourselves to rely confidently on published evidence to inform patient care if the underlying findings might not even be robust.
So open science aims to fix that.
Exactly. The core purpose of open science, therefore, is to address these fundamental concerns. It's best understood as a collection of principles and associated practices aimed at making the entire scientific process, from inception to dissemination, more reliable, more transparent, and more accountable. It's about shifting the culture of science towards greater openness and rigor.
What are some of those key practices?
Key practices within the open science umbrella include things like the wider sharing of research materials, data and analysis code, enabling others to actually verify findings, check analyses, and build upon previous work more easily. It also involves placing greater value on replications and reanalyses of existing work, treating them as important scientific contributions rather than solely prioritizing novel findings.
Mm-hmm.
There are also calls for significant changes in statistical approaches. For example, more thoughtful consideration of statistical power before starting a study, and more transparent ways of presenting data, perhaps showing the raw data points rather than just summary bars. Furthermore, practices like using preprints, making research manuscripts publicly available online before formal peer review, and promoting open access publishing are integral to ensuring that research findings are freely and widely available to everyone, not locked behind paywalls. Ultimately, open science seeks to create a research ecosystem where findings are more robust, more verifiable, and therefore more trustworthy and ultimately more impactful for guiding clinical practice and improving patient care.
That's a huge systemic shift in approach. Among these various practices, pre-registration and especially registered reports, or RRs, seem to be some of the most talked about and potentially transformative changes. Could you explain in a bit more detail what these entail and how they're specifically designed to address the issues we've discussed, like publication bias towards positive results?
You're absolutely right. Pre-registration and registered reports are arguably two of the most significant and potentially game-changing developments within the open science movement, directly tackling some of the core problems we've discussed.
So what is pre-registration exactly?
Pre-registration, in its basic form, involves publicly declaring your research plan, including your specific hypotheses, your study design, your primary outcome measures, and your planned analysis pipeline before you collect any data. You typically lodge this plan in a public, time-stamped registry.
What does that achieve?
It creates a clear, verifiable record of your intentions before you know the results. This makes a crucial and transparent distinction between confirmatory hypothesis testing, testing the specific ideas you set out to investigate, and any exploratory analyses you might perform later, perhaps based on patterns you observe in the data.
And registered reports take it further.
Yes, registered reports. Registered reports take this concept a significant step further by integrating it directly into the journal peer review process. In an RR, you submit your detailed study protocol, your introduction, hypotheses, methods, and detailed analysis plan to a journal for peer review before you collect the data.
Peer review before the study is done.
Exactly. The reviewers assess the importance of the question and the rigor of the proposed methodology. If the protocol is accepted in principle, based on the quality of the question and the methods, the journal essentially commits to publishing the resulting paper, regardless of the outcome. Whether your hypotheses are supported or not, whether the results are statistically significant or null, positive or negative, as long as you follow the approved protocol and the conclusions are justified by the data, the paper will be published.
That sounds revolutionary. It completely removes the bias towards positive results, doesn't it?
It largely does, yes. That's the power of these approaches. They directly combat questionable research practices that contribute to bias. By fixing your hypotheses and analysis plan beforehand, pre-registration makes it very difficult, if not impossible, to engage in things like hypothesizing after the results are known, or arcing, where researchers might notice an unexpected finding in their data and then retrospectively frame it as if it were a pre-planned hypothesis.
Right, fitting the story to the data.
Precisely. It also significantly reduces the potential for p-hacking or selective reporting, manipulating analysis choices, or only reporting favorable outcomes to achieve statistically significant p.05 results. These practices heavily contribute to biased outcomes and the replication crisis.
And the pre-data review and RRs.
The pre-data peer review inherent in registered reports can also significantly improve the quality of experimental design and methodology. Reviewers can spot potential flaws, suggest improvements to the protocol, or demand higher statistical power before the researcher has invested significant time and resources in data collection. This leads to higher quality, more robust research from the outset.
Is there evidence this actually works? Does it lead to more null results being published?
Yes, we have compelling evidence now that RRs are making a real difference in increasing the reliability and transparency of the published literature. A survey looking at the outcomes of hypotheses tested in published registered reports found a really striking figure. 60.5% of the primary hypotheses were not supported by the experimental data.
Over 60% were null findings.
It's essentially, yes, null or non-significant findings for the main pre-register hypotheses. Now, contrast that with the traditional literature, where estimates suggest typically only 5% to 20% of published papers report null findings for their main hypotheses.
That's a massive difference.
It's a dramatic difference. It strongly suggests that RRs are effectively counteracting publication bias and genuinely increasing the chances of important null findings being published. This offers a much more accurate, less biased reflection of the scientific inquiry process. For you, the clinician, this is crucial. It means the published literature, at least the portion using RRs, becomes a far more trustworthy guide. It gives you a clearer, more balanced picture of an intervention's true effectiveness rather than just its perceived successes filtered through publication bias.
That 60.5% figure for unsupported hypotheses is truly powerful. It really lays bare the extent of the bias in the traditional system. It's clear why these approaches are generating excitement. For medical researchers, perhaps orthopedic surgeons listening who are keen to embrace these principles, what practical tools and new systems are actually available now to support these open science practices? Is the infrastructure there?
Yes, the good news is that there is a rapidly growing ecosystem of tools and platforms designed specifically to facilitate open science practices, making it much more practical for medical researchers to adopt them.
Like what, for instance? How can they share their work more easily?
Well, for sharing code and documentation, we now have excellent web-based, version-controlled repositories like GitHub or Bitbucket. These allow researchers to transparently share their analysis scripts, tracking every single change made over time. This means someone else can see exactly how the data was analyzed. Complementing this are scripting formats like R Markdown or Jupyter Notebooks, which allow you to integrate your code, the resulting output like graphs or tables, and explanatory text all in one reproducible document.
So others can rerun the analysis exactly.
Exactly. These tools ensure that when you share your orthopedic research, others can actually recreate your analytical steps precisely, which builds enormous confidence in your findings.
What about standardizing data? That often seems like a hurdle.
It is, but there's progress there too, with growing standardization efforts. A great example, though, from neuroscience is the Brain Imaging Data Structure, or BEADS. It standardizes how neuroimaging datasets are organized and described. This makes them much easier to share, aggregate, and analyze across different labs and studies. We're seeing similar moves towards standardized data formats and reporting guidelines in other fields, including efforts within orthopedics, which reduces heterogeneity and improves the comparability of studies crucial for robust meta-analyses.
Are there tools to check the quality of existing research?
Yes, some analytical checking tools are emerging, although they need careful interpretation. Tools like P-curve analysis attempt to estimate the presence and degree of publication bias, or p-hacking, within a specific body of literature. Others, like StatCheck, can automatically scan papers for inconsistencies in reported statistical values, like p-values not matching test statistics. These can help researchers critically appraise the integrity of the published literature they rely on.
And accessing data itself. Open data.
This is a huge one. There's increasing access to open data repositories. Resources like the UK Biobank and Biomedicine provide researchers with free access to vast, high-quality datasets on hundreds of thousands of individuals. These can be reanalyzed to test new hypotheses or validate findings, democratizing access to valuable research material that might otherwise be incredibly expensive or impossible for smaller research groups or those in less resource settings to collect themselves. This is invaluable.
That's fantastic for researchers without huge budgets.
Absolutely. And finally, we're seeing the growth of collaborative networks, distributed lab networks like the Psychological Science Accelerator or consortia like the INEBJMA Consortium in Neuroimaging. These facilitate large-scale international collaborations, often using standardized protocols and crowdsourced data collection, allowing for highly powered studies that would be impossible for a single lab to conduct. This model holds huge potential for orthopedic research, too, for large multi-center trials.
And new ways to publish this work.
Yes. Alongside RRs, we're seeing new publishing formats emerge. like secondary RRs, where you pre-register analyses of existing data, or exploratory reports, which provide avenues for publishing well-conducted exploratory work transparently, acknowledging its nature upfront.
So lots of tools emerging. What's your practical advice for someone wanting to start?
My practical advice would be this. Try to adopt as many open practices as feel feasible for you and your specific project. Don't feel you have to implement every single one perfectly from day one. Be strategic. Select the methods, maybe start with sharing your data or pre-registering your next study that best fit your research question, your resources, and the stage of your project. And remember the adage, don't let the perfect be the enemy of the good. Even small steps towards greater openness and transparency contribute significantly to improving the overall quality and reliability of medical research.
That's very helpful, practical advice. Start where you can. Now, while open science clearly offers undeniable benefits for the rigor and reliability of research, it's also clear that such a fundamental shift isn't without its difficulties. Let's talk honestly about the challenges. How might these new practices, particularly the structured approaches like pre-registration and registered reports, actually restrict a researcher's flexibility or even feel counterintuitive to the way science has traditionally been done?
You've hit on a significant point there, and it's important to be realistic about the challenges. While hugely beneficial for the quality of science, adopting open science practices, particularly things like pre-registration and registered reports, does impose certain restrictions on the traditional scientific process. And these can feel quite challenging, sometimes counterintuitive, and even frustrating for researchers used to working differently.
What's the main restriction people feel?
I think the most prominent one is the demand for fixed timelines and the strict separation it enforces between exploratory and confirmatory analyses. Pre-registration and RRs require you to publicly commit to your specific hypotheses and your detailed analysis pipelines before you collect or analyze the data.
Which sounds good for rigor.
It is good for rigor, absolutely. It prevents bias from creeping in by tailoring analyses to fit results. However, the flip side is that it means any new insights you gain during the study, maybe unexpected patterns you observe in the early data, or perhaps crucial subsequent publications by others that shed new light on your field, cannot legitimately be used to inform or change your confirmatory hypotheses or primary analyses within that pre-registered study.
So you can't adapt your main plan based on new learning.
Not for the pre-specified confirmatory part, no. You can and should report unexpected findings or conduct exploratory analyses, but they must be clearly labeled as such as post hoc and exploratory. This can feel quite constraining for scientists who are used to a more iterative process of thought and discovery happening throughout their experiment. It challenges that inherent flexibility of scientific inquiry where hypotheses might naturally evolve as data comes in.
And what about mistakes? Does openness make them more problematic?
Well, the increased transparency of open science, sharing data, sharing code, pre-registering plans, inevitably means that mistakes are more visible. If there's an error in your code or a flaw in your pre-registered design, it's out there for others to potentially see. Now, scientifically, this is ultimately a good thing. It encourages greater rigor and allows for correction.
But personally challenging.
It can be. It removes some of the, perhaps comfortable, flexibility to avoid acknowledging mistakes that might have existed in more closed traditional approaches. Researchers must be prepared for their errors, even honest ones, to be more openly discussed or identified. This also means there can sometimes be greater ramifications for mistakes. The loss of flexibility in responding to errors discovered midstream, especially with the fixed timelines involved in registered reports, can mean that a methodological oversight identified later in the process might have larger consequences for the project's publication prospects under the RR model than it might have in a more traditional workflow where analyses could potentially be adjusted post hoc, though often inappropriately. It really requires meticulous planning and piloting from the very outset, which is a significant shift in mindset and workload for many orthopedic researchers who might be more used to refining protocols on the fly based on early results or practical challenges.
That increased visibility of errors and the strict timelines required for planning are certainly a departure from established norms and sound like they can add considerable pressure. Beyond this impact on intellectual flexibility, what are the practical implications of open science for busy medical researchers, especially regarding the sheer time cost involved? Does it take longer?
The time cost is a very real and often significant challenge, and we shouldn't underestimate it, especially for medical professionals who are frequently juggling demanding clinical responsibilities alongside their research activities.
Yeah.
In our experience, and what researchers consistently report, is that adopting these additional requirements of open and reproducible science often consumes significantly more time than traditional approaches.
Where does the extra time go?
Several places. For instance, properly archiving, thoroughly documenting, and quality controlling your code and data so that it's genuinely understandable and reusable by others that takes considerable, often unglamorous effort. But perhaps the most time-consuming aspects are often the formal pre-registration or particularly the registered report processes themselves.
The pre-study work.
Exactly. This involves potentially extensive piloting to ensure your methods work, developing highly detailed, complete analysis pipelines before you even start collecting the main data, writing a comprehensive Stage 1 manuscript, the protocol for an RR, and then undergoing potentially multiple rounds of peer review on that protocol prior to even starting the main experiment.
Wow, all that before data collection.
Yes. All of this happens before you get to the stages we traditionally think of as the bulk of the work, the actual data collection, the final analysis, and writing the final manuscript, Stage 2 for an RR. When you add all these upfront steps together, in many cases, these additional requirements can easily double the overall duration of a research project compared to a more traditional workflow.
Double the time. That's huge.
It can be, yes. Furthermore, as we touched on, open science often rightly emphasizes the need for adequate statistical power, especially if aiming to publish potentially null results confidently. Open experiments, particularly RRs, often require higher statistical power than has sometimes been traditional. This typically translates to needing larger sample sizes.
Which means more time recruiting patients.
Precisely. For a clinical trial in orthopedics, for example, aiming for high power might mean needing to recruit significantly more patients for a study comparing two types of joint replacement and potentially following them up for longer periods. This is inherently more resource-intensive, takes longer, and impacts patient pathways and clinic resources.
And how does this affect researchers' careers?
This increased time cost has a direct and often problematic impact on productivity metrics. For early career researchers, think of residents doing research projects, clinical fellows, or postdocs on fixed-term research contracts completing fewer projects within a given time frame can clash severely with traditional academic metrics of success. These metrics often prioritize the sheer quantity of publications, especially first author papers.
So they look less productive.
Exactly. It can place them at a distinct disadvantage when competing for limited grant funding, fellowships, or permanent academic or clinical academic positions if the institutions or funders evaluating them don't adequately recognize and reward the extra time and rigor involved in conducting open science. I've seen many bright, enthusiastic early career researchers really struggle to balance the scientific desire to adopt these better practices with the pragmatic need to build their publication record quickly for career survival.
So it's not just about individual researchers needing more time, but about the entire academic incentive structure needing to adapt to value these practices properly. That sounds like a considerable hurdle. What are the specific issues with this incentive structure currently, and why isn't it quite ready or fully supportive of open science yet?
You've precisely identified the core of the problem. The current incentive structure within academia and medicine, how we evaluate researchers, award grants, make hiring and promotion decisions, has simply not yet fully caught up with the demands or indeed the benefits of open science. There's a significant mismatch.
In what ways is it lagging?
Well, there's a significant lagging recognition in formal evaluations. Major assessment frameworks like the Research Excellence Framework, REF here in the UK, or internal university evaluation systems are still predominantly geared towards traditional metrics. They focus heavily on the number of publications, the perceived impact factor of the journals they're published in, and grant income generated.
And they don't value open science practices explicitly.
Not explicitly or consistently enough. They don't yet fully endorse or adequately reward the substantial efforts involved in open science practices, things like meticulous data sharing, the time spent on pre-registration, the value of publishing important null results, or conducting rigorous replication studies. These often invisible efforts don't fit neatly into the old metrics.
What about journals and funders themselves? Are they fully on board?
While many are supportive in principle and things are changing, we still encounter journal and funder skepticism in practice. Some reviewers, journal editors, and grant panel members remain unconvinced about the value, or perhaps are unfamiliar with the nuances, of open science methods. They might apply the same critical lens to open science submissions, maybe criticizing a null result from an RR, or questioning the time requested for data curation without fully accounting for or appreciating the substantial extra effort, rigor, and time invested. This means the sacrifices researchers make in terms of project speed or flexibility aren't always proportionally valued or rewarded during peer review.
And the bias towards exciting results.
That absolutely persists. The good story bias is still very much alive, particularly in high-profile so-called glamour journals. These publications often still demonstrate a strong preference for a compelling narrative, typically involving positive, novel, perhaps even counterintuitive results. The more rigorous, careful, outcome-independent nature of open science, with its inherently higher likelihood of reporting null or incremental findings, might not always align easily with this preference for breakthrough stories. This can make it harder, paradoxically, to publish important but perhaps non-flashy open science research in the very top-tier journals that career progression often depends on.
This must disproportionately affect junior researchers.
Absolutely. These systemic factors create particular and quite acute challenges for early career researchers, ECRs. As we discussed, the increased time cost often means they simply have fewer publications on their CVs during critical early stages when they're applying for jobs or fellowships. The strong move towards more collaborative, team-based science, which open science often fosters, can also lead to authorship dilution. While great for science, having fewer first or last author credits can be problematic when evaluation systems still heavily weigh these specific authorship positions.
And pressure from seniors.
That can be another factor. Senior colleague expectations can sometimes create tension. Senior researchers, who perhaps built their careers using quicker, more traditional methods, might still expect similar levels of publication output from their junior colleagues or trainees without fully appreciating the additional time and different outputs associated with rigorous open science. And finally, the very high power standards required by some journals for registered reports, some demand 95% power, can be extremely challenging or even prohibitive for resource-intensive clinical studies, like many in orthopedics. This could potentially skew which research areas or questions are deemed feasible to conduct using these best practice methods. All these systemic disincentives combine to make the adoption of open science a brave, scientifically laudable, but often professionally difficult choice for individuals, especially those just starting out.
It sounds like a really complex landscape where individual commitment to better science can genuinely clash with the prevailing systemic inertia and reward structures. It highlights the need for change at an institutional level, not just individual effort. But let's shift to a more optimistic outlook now. Despite these significant challenges we've just discussed, what are the compelling long-term benefits that make investing the time and effort in open science a truly worthwhile endeavor, both for individual researchers and for the advancement of science as a whole? For our listeners, why should they make this investment despite the hurdles?
The long-term benefits are indeed compelling, and I believe they significantly outweigh the short-term challenges. They point towards a future where medical research is fundamentally more robust, more efficient, more collaborative, and ultimately much more impactful for patient care.
How does it benefit individual researchers directly in the longer term?
Well, for individuals, embracing open practices like sharing data and code or using preprints often leads to increased acknowledgement and collaboration. Researchers can actually receive citations and credit specifically for their shared datasets or software, not just their final published articles. Preprints increase the visibility of work much earlier and can generate valuable feedback and opportunities for collaborative projects that might not have happened otherwise. This fosters a more connected and supportive research community. Imagine more collaborative, multi-center orthopedic trials emerging from shared protocols or early findings.
And for the progress of science itself?
For science as a whole, open science makes preclinical and translational research demonstrably more robust and efficient. By reducing the waste associated with irreproducible findings or duplicated effort, and by improving the reliability of the evidence base, it can genuinely accelerate the pace of discovery and the development of effective, life-saving drugs, diagnostics, and innovative medical devices or surgical techniques. Think about the rapid development and testing of vaccines and treatments during recent public health emergencies. Open science principles like rapid data sharing and collaborative platforms played an absolutely vital role in that acceleration.
Does it make research more widely read or used?
There's good evidence for that too, yes. It leads to wider reach and impact. Multiple studies have now shown that articles that make their data openly available tend to receive significantly more citations than comparable articles that keep their data private. Similarly, using preprints and pre-registering studies, because they make the work publicly accessible much earlier, also tend to increase subsequent citation rates and can lead to earlier media coverage or policy uptake, amplifying the impact of the research much sooner and more broadly.
You also mentioned equality earlier.
Yes, and that's a crucial point. Open science can play a significant role in promoting equality and democratizing research. By making resource-costly data, perhaps complex imaging data sets, large genomic sequences, or extensive clinical trial data openly accessible, it allows researchers in less funded institutions or those in low and middle-income countries to engage with high-quality research material without needing multi-million-pound grants to collect it themselves. This fosters wider participation, brings in diverse perspectives, and ultimately enriches the global scientific endeavor. It helps level the playing field. This creates a virtuous cycle of improved knowledge generation and application.
Those are certainly powerful incentives for the entire scientific community, promoting efficiency, impact, and equality. But bringing it back to the individual clinician researcher, perhaps an orthopedic surgeon balancing clinical work with academic pursuits, how do these broader benefits translate into tangible career advantages for them, especially those navigating their earlier mid-career stages? Can it actually help them get ahead?
Yes, I strongly believe it can, and increasingly so. For individual medical professionals, especially those looking to build sustainable and impactful careers in research, early adoption of open science practices can confer significant, tangible advantages in the evolving landscape of academic medicine and beyond.
How so? What's the direct benefit?
Firstly, and perhaps most importantly in the medium term, it positions you ahead of the curve. There's a growing consensus that open science practices are becoming the norm, not just a niche interest. Regulatory bodies are demanding more transparency, funders are increasingly mandating open practices, and journals are adopting new formats like RRs. Those researchers who proactively developed skills and an established track record in these methods now will have a significant advantage as these practices become standard expectations. It demonstrates foresight, methodological rigor, and a clear commitment to research integrity and best practice.
So it makes them look good to employers or funders.
Exactly. It can make you a more attractive candidate for competitive research grants, sought-after fellowships, senior academic or clinical academic positions, or even leadership roles within your institution or professional society. It signals equality.
You also mentioned industry.
Yes. Secondly, it significantly enhances your industry relevance. Reproducibility, transparency, and data integrity are absolutely paramount in industrial R&D contexts, particularly in pharmaceutical research and biomedical device development. Therefore, for clinicians who might consider career transitions toward industry roles or collaborations with industry partners, having demonstrable open science skills is becoming a highly sought-after and valuable asset. It can genuinely differentiate you in a competitive job market or partnership negotiation.
And are funders and institutions actively supporting this now?
They are, increasingly. Thirdly, there's rapidly growing funder and institutional support. Major grant funders worldwide are not only increasingly requiring data sharing as a condition of funding, but many are also actively offering specific funding streams dedicated to conducting replication studies or developing and implementing open science projects. And crucially, some leading institutions, particularly universities, are now starting to explicitly incorporate open science contributions into their hiring and promotion criteria. For example, some are asking candidates to describe their track record in open science methods or contributions to open resources as part of job applications, even in fields like orthopedic research. This means that embracing these practices isn't just good for science. It can directly improve your prospects for securing funding, getting jobs, and advancing your career.
That's a very compelling case for making the investment now, Professor. It truly sounds like the scientific landscape, particularly in medicine, is undergoing a fundamental and likely irreversible shift towards greater openness. What's your final take, then, on the broader outlook for the future of research as it continues to embrace these principles?
My final take is that the transition towards adopting open science methods more broadly is not just beneficial, it's fundamentally necessary and, I believe, ultimately inevitable. The serious issues highlighted by the replication crisis, the unacceptable levels of research waste, the damaging effects of publication bias, the pervasive lack of transparency are simply too fundamental to the integrity and progress of science to ignore any longer. And there's a powerful momentum building. Once researchers, particularly medical professionals who are driven by the need for reliable evidence, truly grasp the crucial conceptual distinction between confirmatory hypothesis testing and exploratory research, and once they begin to implement open practices like pre-registration, data sharing, and using registered reports, well, in my experience, it's very difficult to revert to the old, less transparent ways of working. The intellectual clarity, the increased confidence in one's own findings, and the contribution to a more reliable evidence base that open science brings are deeply rewarding, despite the extra effort.
So it creates its own positive feedback loop.
Exactly. It creates a powerful virtuous cycle. As more researchers adopt these practices, the overall quality and trustworthiness of the published evidence base improves. This, in turn, benefits the entire medical community, from individual clinicians like yourselves, making critical decisions at the bedside, perhaps about adopting a novel spinal fusion technique based on robust RR data, right through to policymakers shaping national public health strategies based on more reliable meta-analyses. It builds greater public trust in science and medicine, and ultimately it has to lead to better, more evidence-informed patient outcomes. It's undoubtedly a long journey, with cultural and systemic hurdles still to overcome, but it's a journey we must collectively undertake to ensure the integrity and utility of the knowledge you rely on every single day in your practice.
Professor Moimam, this has been an incredibly enlightening and, I think, truly vital deep dive. We've unpacked the staggering reality of unreported clinical trials that hidden half of the evidence. We've explored the broader, concerning landscape of research waste. We've confronted the deep-seated biases against negative findings and the profound ethical implications. And importantly, we've examined how legislative efforts and, crucially, the growing movement of open science, despite its challenges, are paving a path towards greater transparency and reliability in the medical research that underpins everything we do. For all of you listening today, I really encourage you to take a moment and consider how these insights might inform your own engagement with research, whether you're conducting it or consuming it. Think about how embracing transparency and advocating for open practices can strengthen the evidence base you rely on every single day, making a tangible difference for your patients. If you found this deep dive valuable, please do take a moment to rate and share our deep dive with a colleague who might also benefit. And as we close today, perhaps consider this final thought. If every single clinical trial conducted decades ago on the treatments and techniques we still commonly use today were fully transparent and publicly accessible right now, how profoundly might our current understanding of patient care actually change? It's a thought that truly underscores the ongoing importance and urgency of this conversation. Thank you so much, Professor Moimam, for your invaluable insights and expertise today.
It was my pleasure. Thank you for focusing on such an important topic.
And thank you, our listener, for joining us on this critical exploration into the unseen evidence. We look forward to welcoming you back for the next deep dive.